The equine body tells us many things when we listen carefully. Thanks to innovative research, listening as horses recover from injury may soon be possible at the molecular level.
One of the major hurdles that we face as sports medicine and rehab veterinarians is really deciding when that horse might be ready to go back to work or not. And right now, the tools that we use are x-rays, ultrasound, clinical exam, how the patient is doing at a certain level of workload. But we really don't have any sort of objective or numbered data to indicate that we might be right about that.
We're trying to develop biomarkers that we can utilize to monitor horses that are in rehabilitation as another way to objectively monitor them, see how they're doing in that progression.
Thanks to a Foundation for the Horse Innovation and Discovery Grant, Dr. Alan Page and Dr. Sherry Johnson are discovering a novel way to measure a horse's healing and recovery progress. They approach their work from two very different perspectives, one grounded in academic research, the other in hands-on equine rehabilitation. But their collaboration is deeply complementary.
Dr. Page and I are somewhat of an unlikely combination in that he is very academically based out of University of Kentucky with access to all of the fancy rehab equipment. And most of his days, are spent at the research lab doing benchtop stuff. I'm actually in the trenches every single day here in North Texas, working with horses, working in the barn, and diagnosing them, carrying them through their rehab journey.
Dr. Page and Dr. Johnson are studying messenger RNA, the body's early instruction system that indicates what tissue is preparing to do before change is observable.
Messenger RNA, I think, has become a little bit more familiar to people in the last couple of years. It is the recipe for the creation of specific proteins. So rather than measuring those proteins, we're actually upstream in the cell's process of developing those proteins. The nice thing about that is in horses, we can look at upwards of 22,000 or so different biomarkers.
The genesis of this project was really to look at a variety of blood parameters to see in horses already undergoing professional rehabilitation if there's changes or trends that are consistent with healing or lack thereof. And if there's a way we're able to tease out what some of maybe those biomarkers are that indicate healing or a lack thereof. We know that all horses are different. Each athlete is not the same as the next, and every single horse's goal is potentially different. But we don't understand yet physiologically that much about these recovery rates and these tissue-specific responses.
The ability to monitor messenger RNA could drastically change methods and outcomes in horse rehabilitation.
Rehab for horses in and of itself is still very much in its infancy, and there's still so much that we don't know about a lot of the modalities that we're prescribing and a lot of the various tissue-specific responses that we have. This research directly impacts horses, it directly impacts owners, and it really helps us put science behind the recovery itself.
I'm so appreciative of, from the foundation standpoint, from the funding, is that it's what I would certainly consider to be a high-risk, high-reward grant. We didn't have a lot of data coming into this. We obviously have been looking at these different blood biomarkers in other athletic disciplines, but had never done it in something like rehabilitation.
The fact that Coyote Rock Ranch and the Foundation for the Horse were forward-thinking enough to believe in the two of us coming together for the betterment of horse and their health care really speaks to the fact that everyone really is pulling the wagon in the same direction to push the needle forward. So grants like this really help us pave that path forward.
Discovery often begins with uncertainty, and the Foundation for the Horse's Innovation and Discovery Grants are transforming that uncertainty into opportunity to advance the well-being of the horse.